to specific pathogens. So I will explain about mechanism of innate immunity. First line of defense, which is physical and chemical. First point, intake skin and mucous membrane act as mechanical barrier to block pathogen entering the system. Point number 2, body secretion like stomach acid, lysosome, which is tears and saliva, chemically destroy microbes. Second line defense, phagocytic cells engulf and digest pathogens via phagocytosis. Second point, natural killer NK cell destroy infected body cell and cancer cell. Point number 3, antimicrobial proteins, interferons inhibit vital replication while complement lysis bacteria cells. Number 3, inflammatory response. Tissue damage triggers mast cells to release histamine. Histamine causes vasodilation, accelerating delivery of phagocytes to infection site for healing.
So, let's quickly break down the key difference between the first and second lines of defense in our innate immune system. Think of the first line of defense as our body external shield. Located at our boundaries, it acts as a passive barrier by using physical and chemical barrier like skin, mucous membranes, tears, and gastric juice. Best of all, it is always present and active. But what happens when a pathogen breaches this boundary? That's when the second line of defense steps in. Operating internally within our tissue and blood, this is an active, rapid cellular and chemical attack. Using phagocytes, natural killer cells, inflammation and fever, its goal shifts to destroying invaders that have already crossed the line. In short, the first line keeps them out, while the second line destroys them if they get in. Together, they form an incredibly coordinated system that guards us 24 hours.
Now, I will continue about acquired immunity. Unlike our innate immune system, acquired immunity is highly specific. It develops only after we are exposed to a particular pathogen. While it is a bit slower to respond, it is incredibly precise, and it works through two main pathways. The first one is humoral immunity, which relies on B cells and antibodies in our body fluids. Secondly, our cell-mediated immunity, which relies on T cells that directly attacks and destroy infected cells. Secondly, we are going to take a look at antigen recognitions by lymphocyte. To kick off this response, we need antigen recognitions. An antigen is simply any foreign molecule that can trigger an immune response. On the surface of these antigens are specific, localized chemical structures called epitopes. Think of epitope as a fingerprint that our lymphocytes use to recognize exactly what they are fighting. Now we are going to take a look at lymphocytes development. The main players here are the B cell and T cells. Both originate from stem cells in the bone marrow. However, B cells complete the entire maturation process in the bone marrow and are responsible for producing antibodies. T cells, on the other hand, migrate to the thymus to mature, where they become critical for destroying cancerous and infected cells. Interestingly, both types of cells have about 100,000 receptors on their surface, all designed to recognize the exact same epitope. Now we are going to take a look at origin of self-tolerance. An essential safety measure occurs during their development. The origin of self-tolerance. As B and T cells mature in the bone marrow and thymus, their receptors are rigorously tested against our own body molecule. If any lymphocytes happen to be reactive to our own tissue, it is immediately eliminated through a process called apoptosis, or called as programmed cell death. This ensures our immune system does not mistakenly attack our own healthy cells. Finally, this leads to the second immune response, the foundation of immunological memory. If the same pathogens enter our body for the second time, the long-lived memory cells are immediately activated. They rapidly divide to produce a massive clone of lymphocytes, resulting in a much faster, more effective defense than the first time around. This is exactly how vaccine works. They produce memory without making us feeling sick.
Now I'm going to explain the process of cell-mediated and humoral response. First, humoral response or B lymphocyte pathway. Based on the diagram here, a naive B cell recognizes a specific antigen using its B cell receptor. We call it as BCR. The B cell becomes activated, and the activated B cells undergoes proliferation, or we call it as clonal expansion and differentiation. And most of the cells become plasma cells, and that plasma cells secrete antibodies, and the antibodies bind to the antigen, forming antigen-antibody complexes. And these complexes are removed by phagocytes, eliminating the pathogen. In other words, the humoral response fights pathogens outside body cells by producing antibodies. We're going to look at cell-mediated response or T lymphocyte pathway based on the diagram here. An antigen-presenting cell ACPC displays an antigen on an MHC molecule. A naive T cell recognizes the antigen through its T cell receptor, which is TCR, with the help of CD4. And these T cells become activated, then undergoes proliferation and differentiation. And the activated T cells differentiate into helper T cell or CD4+, which release cytokinesis to coordinate and stimulate the immune response. And the cytotoxic T cells or CD8+, which recognize infected cells. And the cytotoxic T cells actually release porphyrin and granzymes, causing the infected cell to undergo apoptosis or programmed cell death. In other words, the cell-mediated response destroys infected body cells rather than producing antibodies. In conclusion, the immune system is essential for protecting the body against harmful pathogens and diseases. All these responses that we have explained work to eliminate infections, coordinate the body defense mechanisms, and provide long-term protection through immune memory. Therefore, understanding the immune system helps us appreciate how the body maintains health and fight diseases effectively. And that's all for us. Thank you!